Hi, my name is Jacinthe Mars. I'm a PhD student at University of British Columbia in the Faculty of Forestry under the supervision of Sue Grayston and Cindy Prescott. I'm here today to do a walkthrough of my poster called It's a Bacterial World, Microbial Diversity in Reclaimed Oil Sands Soils. The reclaimed oil sands soils are situated in northern Alberta in Canada. Canada has the third largest reserve of oil in the world. 20% of that reserve is extractable through open mining techniques. So far, around 800 square kilometers of land have been disturbed to these techniques. However, after land disturbances, companies have the legal obligation to restore previous land capabilities. Here's two pictures of my research soils. The first one is a natural soil situated in the vicinity of the mines. It is actually a naturally disturbed soil that have been through a fire recently. We choose natural disturbed soil to make a comparison with the reclaimed soil, which is right next to it. As you can see, those two soils are quite different. But does that mean that they have different land capabilities? This is the main question I wanted to tackle with my research project. Here I'm going to show you a smaller part of my PhD project, which has to do directly with, or more specifically, with microbial communities. One of the first aims I had in that project was to look at the structure of the communities. So I wanted to know if the reclaimed and the natural disturbed soils host different microbial communities. I also wanted to know if the tree species planted during reclamation has an influence on microbial community structure. To do that, I had 20 research sites, 5 naturally disturbed sites with fire, 5 reclaimed sites planted with coniferous species, 5 reclaimed sites planted with deciduous species, and 5 reclaimed sites planted with grasslands. All the research sites were between 20 to 30 years old. Soil samples were kept cold until arrival in the lab. Once in the lab, I did the DNA extraction using conventional method. I then used PCR amplification techniques to specifically target the 16S genes. This gene will target specifically bacterial and archaeal species. Then, samples were sent out for sequencing. The first results we can see here is a bar graph showing the proportion of sequences classified either as bacterial or archaeal within the total number of 16S sequences identified. The results are in percent. First thing we can see here is that there's more bacterial than archaeal species in the samples. This is a bit surprising since I thought that there will be a bit more archaeal species in my soil samples. Another result we can see here is that natural sites have less bacterial species compared to the reclaimed sites. On the contrary, natural sites have more archaeal species compared to the reclaimed sites. Now let's jump to alpha diversity. Alpha diversity is a measurement of the biodiversity within each samples. Here I've used the inverse Simpson index to represent it. On this graph, we can see the mean and the standard error of this index. The higher the index is, the higher the biodiversity is in each treatment. What we can see is that the reclaimed treatments seems to have a higher biodiversity compared to the naturally disturbed sites. This might be explained by the source of the material used to do the soil. Maybe it was quite diverse, hence a higher diversity in the communities in these soils. However, I have to note that these differences were not statistically significant. I've also measured beta diversity. Beta diversity will compare diversity between sites. Here I've used principal coordinate analysis to represent that. Principal coordinate analysis is a multivariate statistical tool that will plot together sites that have similar microbial communities. Here what we can see is that the first dimension, so the x-axis, separate pretty well the natural sites and the reclaimed sites, showing hence that they have a different microbial communities or a different microbial community structure. One interesting point here is the NS2 sites. NS2 site is quite interesting because it is a site that I have visited the first year that was a natural forest, but then I didn't sample it for microbial diversity. I did it the second year. However, the second year, there were a road on my site, forcing me to take the soil samples just beside the road in the natural forest. So here maybe we see an indication of disturbance on the microbial communities. This is why it is plotted together with the reclaimed sites. What we can also see is that the grassland sites and the deciduous sites are grouped together on this principal coordinate analysis. The coniferous and the deciduous species are plotted together, which means that they're not quite different. So what we can see here is that the natural and the reclaimed sites do have different structure of microbial communities, and that tree species, namely deciduous and grassland sites, have different microbial community structure. I've also plotted the communities that were significantly correlated with the axis on this graph. I didn't do a thorough analysis of these communities, but what I can see is the communities that were found in the naturally disturbed soils were communities that were usually found in abundance in natural soils. However, in the reclaimed soils, there were few communities that are associated with the methane cycle. These communities do transform carbon into methane. Maybe because of the machinery used during the construction of these sites, they have compacted the soil, created area with less oxygen, and hastening the production of methane in these soils. Taken together, these results showed that the microbial community structure do differ between reclaimed and naturally disturbed sites. They also showed that the communities differed between grassland and deciduous reclaimed soils, supporting the fact that vegetation does have an effect on microbial communities in soils. We also shown that the alpha diversity, so the biodiversity, is not significantly different between the reclaimed and the restored soil, but seems a bit higher in the reclaimed soils. I would like to thank everyone who helped me on the field and in the lab. Also, thank you for listening. If you have any question, don't be afraid to email me. I will answer any question you have. You can also reach me through any matters, and I'll be very happy to answer all your questions.